Good evening, everybody. So it's really awesome to see so many people in this auditorium again for another season of Friday evening lectures. I know we started last Friday, and really excited to pick up some science here tonight. But first, just a few housekeeping items. Well, first of all, I'm Sebastian Lay, MBL's Chief Advancement Officer, and my main purpose tonight is to just ask you a few things and tell you a few things. First, please turn off or mute your cell phones. As somebody once said, turn them to stun. You know, that can help. Secondly, please just know at the end of the talk tonight, you'll have a chance to ask questions in the audience and from online. And so if you're in the audience, please just wait for a microphone so that the folks that are online can hear the question. And then there is a reception that follows over on the Swope Terraces, and that'll give you an additional chance to talk to this evening's speaker. And then finally, just to let you know that tonight's lecture is being simulcast and recorded. So that's an obligation of the state of Massachusetts to let you know that. And finally, welcome. And I know you'll continue the conversation after we finish this evening, but this is over 135-year-old tradition or so. So no pressure to keep the intellectual discourse and the inquiry going, but I know we'll be off to a roaring start with our speaker tonight. So, and then our introducer tonight is my boss, the director of the MBL, Dr. Nipam Patel, who knows our speaker well. So thank you.
Thanks, everyone. It's a great pleasure to welcome you. As Sebastian said, it's great to be able to do these things in person. And welcome to the second of the series of Friday evening lectures for this summer. So this lecture is known as the Glassman Lecture, and the Glassman Lecture is held in honor of the late Harold N. Glassman, who left a generous bequest to the MBL, and the funds from that bequest are what allow us to have this lecture every year. So I'd like to thank my appreciation in all these kinds of instances to people who generously support the MBL in this way. And the Friday evening lecture is one of the great ways that we try to give back to the community. So I'm very happy to introduce today's speaker, Scott Edwards, who I've known for a number of years. So Scott is the Alexander Agassiz Professor of Zoology and the curator of the Ornithology Museum of Comparative Zoology at Harvard University. And so he's an evolutionary biologist, but he uses all sorts of tools to accomplish his work. So he's really known for combining incredible field work where he travels around the world looking at birds, and birds is the system that he works on. But he combines that with a really deep understanding of how to use molecular tools. So a lot of you may know, a lot of effort has been spent sequencing lots of animals. And what do we make of that? But I think Scott's work is a great example of how to take that kind of information, combine it with a really fascinating interest in organisms out in the real world to understand that interplay between their DNA sequence and what that tells you about how they live their lives. And so I'm very excited to hear about his latest results in that area. So Scott has, a number of incredible honors. He's an elected fellow of the American Association for the Advancement of Science, a member of the American Academy of Arts and Sciences, and a member of the U.S. National Academy of Sciences. He's also spent time in the public service he was the director for the Division of Biological Infrastructure at National Science Foundation for a while. And so Scott's, been an incredible influence in the area of both evolutionary biology and genomics. So without further ado, I'll turn it over to Scott.
Thank you, Nipam. It's great to be here. So many people in person. This is awesome. And for all of you folks online, glad you're there as well. As Nipam mentioned, I am an evolutionary biologist, and we finally got around to our lab photo after a couple years of COVID. You can see it's not too long ago. But I've got a really wonderful crew that studies all things avian. Everything from it's covered here, phylogeography. We're interested in how organisms have spread across the globe and why they're found in the places where they're found. And we look at ecological genomics. In this case, we're really interested in interactions between hosts and pathogens and diseases, and how do diseases cause evolutionary change in bird populations. And what I'll focus on today is really in this sector here, in the area of comparative genomics. How can we compare different species phenotypes, the way they look and sound, and their genomes as well? How can we take that information to understand the genetic basis of the amazing diversity that we see in the biological world? Now, I am really fortunate to work in a natural history museum. The Harvard Museum of Natural History is the public side. Many of you have probably been there. The Museum of Comparative Zoology is sort of the teaching and research side. And there we have amazing collections of all branches of the tree of life. And, museums have been, we got to take care of them. In the past few years, I participated in a number of efforts to sort of raise the profile of museums and remind the public of how important they are in terms of everything from disease surveillance to forensics. And so there's a lot of amazing stuff in the museum. And there's, of course, an interesting connection with MBL. Some of you who've been to the library here at MBL might recognize this quote from Louis Agassiz, the founding director of the MCZ. Agassiz was a Swiss geologist, and he sort of a swashbuckling came over to America and founded this museum. And I actually take this quote to heart. A lot of our work is the questions we ask are generated in the field, observing birds, looking at their natural history. And in this era of genomics, it's increasingly a challenge to get students off their computer terminals and into the field. That's not an easy thing to do. But so this is a great quote. Worth mentioning as well that Agassi, there are a lot of skeletons in the closet in terms of issues of domination and compromising indigenous peoples. And in this case, you might have heard of a very sort of a troubling set of photographs that were housed in the Peabody Museum, which is the anthropology wing, of slaves, which Agassi took in his effort to rank human races. And even at the time Agassi was working, he was well known as a pretty virulent racist. And so I would say museums are incredibly useful, but we also have this challenge that we're facing now and that we're trying to work out. How can we decolonize our mission? How can we make museums more inclusive? And so that's a challenge, and that's something where we need input from everyone and how to open our doors and make everyone feel welcome. So today we're going to talk about how can we use the tree of life to understand the genetic basis of traits. This is a review article that I wrote with a couple colleagues, and we were all at a meeting, this meeting of the Society for the Study of Evolution. It sounds a bit like a satanic underground society, but it's actually a very vibrant group of people. And in fact, I'll be heading there tomorrow. And phylogenies are evolutionary trees, and most of the effort that folks have put into using them to understand evolution has gone into fields like biogeography or diversification. How have species come to multiply across the Earth? These are really important questions. And we realized that, well, actually, phylogenies are good for lots of other things as well. In particular, for thinking about novel adaptations and how can we understand the genetic basis of traits. And so we wrote this article to sort of help jumpstart this new use for phylogenies. And so today I'll be talking about a couple examples where we can use comparative biology to understand such diverse things as how birds taste sugar, or where do feathers come from? What parts of the genome are underlying feathers? And in the end, I'll talk about the loss of flight in birds. And so it's a very exciting time right now, and we're just, I think, scratching the surface about how useful these phylogenetic trees will be. And I went to graduate school at UC Berkeley, and was fortunate enough to work in the lab of Alan Wilson, who was, in his day, really one of the most famous molecular evolutionists working. And he and Mary Claire King, she's a professor of genetics at University of Washington, they published this seminal paper in which they basically compared the proteins of humans and chimpanzees. And this is at a time when doing these comparisons was really, really difficult. Sequencing a protein was just arduous, but they marshaled all kinds of data together to basically point out and measure the protein differences between humans and chimps. And the number that they came up with in this paper from 1975 is still holds, and that is that humans and chimps differ basically by about 1% to 2% in their protein sequence. Now, Alan was a master at turning a sort of a negative result into gold. A graduate student might say, oh, I only found one difference out of 100 in these proteins. What am I going to do? Well, Alan and Mary Claire King realized, well, what this means is that, well, it's just not the proteins that are causing the differences in behavior and the phenotype between humans and chimpanzees. Rather, it's probably the way in which those genes are deployed when they are expressed and how they are regulated. This is the whole field of gene regulation. So that's the fundamental distinction that I want to draw for you, is that we can talk about differences in sequence of proteins, and I will show you some really cool examples of that. But we can also talk about the where and the when of gene expression, a phenomenon which doesn't necessarily involve any change in the sequence of the genes that are being expressed. So it's really that dichotomy of, are the proteins changing, or are the genes underlying those proteins simply being deployed at different times and places during development? And that's a really important question in evolutionary biology now. Is it gene sequence change or gene regulation change that is driving evolutionary change? So the first example I want to tell you about involves how birds taste sugar. So we know that we have taste receptors on our tongues, and it's a very small multi-gene family, only a few different members. But you can see these are what we call heterodimers. They're basically two subunits, one of which allows us to taste umami, or savory taste. Think amino acids, or basically meat, for example. And then there's another pair of these receptors which allow us to taste sugar and carbohydrates. And these are spread out all over our tongues, and we have some amazing natural experiments that evolution has performed for us. And so, for example, these pandas, whose diet is basically 100% bamboo, they have no use for a umami receptor. They don't eat meat. And so as a result, this gene has been deactivated and lost essentially from the panda genome. Now, cats, coming from a long line of carnivorous predators, despite the fact that they love milk, for example, they don't have a sweet taste receptor, and because of this high level of carnivory. And so, Darwin's adage, if you don't use it, you're going to lose it, that definitely holds true for the taste receptors. Now, birds are a really interesting case because they too evolved from a highly carnivorous lineage of theropod dinosaurs. Think T-Rex, Velociraptor. This, of course, is Archaeopteryx, one of the most famous fossils discovered. Amazing feathers all over its wings and body. And so it turns out that if you look in the genomes of birds, you can't find a sweet taste receptor. It was lost in the common ancestor or sometime before. Even in species such as hummingbirds, which we know, of course, can taste sugar with exquisite efficiency. I mean, it only takes a hummingbird a millisecond to realize whether this clear liquid is sugar water or clear water. So clearly they are able to taste sugar, and yet they don't have the sweet taste receptor. So how are we going to figure this out? This is the question that my graduate student, Maud Baldwin, endeavored to answer. And, I have a big sort of practical streak in me, and I just said, Maud, this is going to be impossible. Do something simpler, please. Fortunately, she had the persistence and the stubbornness to ignore my advice. That's actually the advice I give my graduate students. Don't listen to your advisor. But she persisted and was able to, in a very kind of phylogenetically focused way, she looked at hummingbirds, of course, but also the closest relatives of hummingbirds, the swifts, and an outgroup of chicken. And in this trio, it's only the hummingbird that can actually distinguish sugar water from plain water. But what Maud was able to do was she was able to actually express these receptors of these species in a cell system that allowed her to basically interrogate what compounds were these receptors responding to. And her candidate receptor, the one she first started to look at, was in fact the umami receptor. Why not look right next door? Maybe something interesting has happened there. And remarkably, she was able to show, so she visited a lab in Japan, and it turns out this lab was basically an arm of the Kikkoman soy sauce company, and they were the only lab in the world that could actually, could do these exquisitely sensitive experiments, expressing taste receptors, finding out what made them tick and what caused them to fire. And she basically discovered that, in fact, the umami receptor, the one focused on meat, had evolved, its sequence had changed such that it could now bind sugars and carbohydrates. So it was a dramatic example of protein evolution causing a new functionality and probably behind the remarkable radiation of hummingbirds into over 300 species. So this was a point in favor of the really contradicting Wilson's hypothesis that it was gene regulation. This is a clear example of the protein sequence changing, causing a new functionality. Maud's gone on to look at this question in even further detail. She first basically cataloged all the different diets all across the bird tree. And what she was surprised to find is there's actually a huge number of species in this clade called the songbirds. It's a group of, we call the Oscine passerine birds, about 4,000 species. And there's a high incidence of nectarivory and ingestion of sugar. And she was able to, and here's some examples of some of the diverse lineages that are nectar feeding, birds from Hawaii and other kinds of nectar feeding species. And she was able to ask whether the ability to taste sugar in this clade was different or the same as that in the hummingbird clade. And she did a remarkable series of experiments where she mixed and matched the subunits of hummingbird umami receptor and that of one of the passerines, in this case a honeyeater. And she showed that, in fact, when you mix and match these subunits, when you work on the native subunit, you actually get a very strong response. So they're definitely binding sugar. However, when you mix the two sets of subunits from different species, that receptivity goes way down. And what that means, of course, is that these two subunits have co-evolved. They're working together to cooperatively to bind to detect sugar. And it also strongly implicated that this ability to taste sugar in these songbirds was an independent evolution of that ability. It didn't descend from the same set of receptors that hummingbirds use. She was able to refine this even more. As we look across this tree, we can see a bunch of Oscine songbirds here. And then we have a couple species called the sub-Oscines. These are largely neotropical species. And she was able to, again, through these swapping experiments, was able to show that whereas when the subunits from the sub-Oscines were mixed with those of the honeyeater, you don't get much of a response to binding sugar. However, various subunits from various species of Oscines could work cooperatively to elicit a very strong sugar reception response. And what this says, again, is that these two subunits are working together, and it shows that this evolution of taste of sugar perception in the songbirds is probably a single origin, but that it happened very early in the diversification of this clade. And so, again, another really interesting example of protein change causing evolutionary change. Now, you're probably familiar with the genome of vertebrates, including the human genome, is incredibly complicated. Of course, the human genome is about 3 billion letters. Those of birds, such as ones I study, are typically about anywhere from half that to a third that, so they're often about 1 billion letters. Now, most of those letters, in fact, don't encode proteins. I mean, I think many of you appreciate that the human genome is only about 2% comprised of actual genes. And so what's the other 98% of the genome doing? Appropriately referred to as dark matter. It's all over the place, but we don't know what it does necessarily. And one hypothesis is that it's a vast sort of regulatory network. In other words, 2% of the genome is actually sort of the business end, the enzymes and the proteins with their functions, but maybe the other 98% is orchestrating the deployment of those proteins and genes. And we have sort of taken this very complicated landscape and focused on one particular foothold of that, which we call these conserved non-exotic elements. What this means is basically it's a part of the genome that is highly conserved between species, but it doesn't encode a gene. It's somewhere between genes, or maybe it's in an intron or something like that. This is very intriguing, and that's indicated by these black, this solid region showing high sequence similarity between mammals all the way down to fish and frogs in a region that's completely outside of a gene, okay? Now, in molecular evolution, when you see this high conservation, that means that that region is doing something important. It's actually a very powerful test for determining whether a region has an ongoing function. We may not know what that function is, but the fact that it's stayed the same for so long tells us that it's doing something really important. And one of the things scientists have learned in recent years is that some of these non-coding conserved elements may act as what we call enhancers. Enhancers are parts of the non-coding genome that essentially recruit the machinery that is going to allow genes to be expressed. So you can see here all these things called transcription factors are coming together with these replication enzymes and basically facilitating gene expression. Sometimes enhancers work on genes that are quite far away in the genome, sort of long distance regulation. But this whole phenomenon of the role of enhancers in gene regulation is extremely interesting. And it's one way in which scientists have begun to understand the very complicated and unusual language of the non-coding genome. I mean, we understand proteins, right? That's the triplet code. That's, you know, it's very, very straightforward. We get that. It was discovered in the 60s. The language of the non-coding genome is, it's sort of like Ouija board genomics. It's very, you feel kind of weightless. You're disoriented. You don't know how it's actually working. So let's do an experiment where we're looking at the relative role of genes versus gene regulation in the origin of a very iconic trait in birds, namely feathers. Now, we've learned over the past couple decades that feathers, in fact, don't define birds. Feathers are well known in the fossil record long before the first birds appeared. Remarkable fossils from China and other places showing clear sort of proto-feathers, if you will. Not so much the branching structures that modern birds have, but sort of monofilaments. And then, of course, we have Archaeopteryx with this extraordinary flight feathers, feathers on the tail, on the long bony tail. Scientists can even tell you the color of these feathers by looking at the size and shape of these organelles, which you can see by electron microscopy called melanosomes. And so we have really data-based evidence for coloration in a lot of these extinct bird ancestors. But what we did, we wanted to know what caused feathers to evolve in the first place. So what we did was we went through a catalog of about 400 papers. This was a kind of experiment where we actually, we were just working on existing data. We didn't actually generate any new data ourselves. But we wanted to catalog all of the genes that developmental biologists had told us were important for the development of feathers, because those are the genes that are probably important for the origin of feathers. We actually found almost 200 genes, which was a lot more than I had expected. Now, around those 200 genes, as you see in this diagram below, we've got a lot of these conserved non-exonic elements. And so, for example, here's a gene called SHOX. You can see the exons here, these red squares. Now you can see that those exons are conserved all through all these species. This is an alignment of different parts of the genome of a variety of birds and mammals and all the way to fish. So you can see that SHOX is in fact likely present in the common ancestor of all of these vertebrates here, okay? Because we can find the signatures of the exons in those genomes. Now, by contrast, a lot of these non-coding elements have arisen at variable times in evolution. We've color-coded them here so that you can indicate where in the tree of life they arose. So the red ones are bird-specific. They're only found in bird genomes. Here's one in green, which is found in birds and reptiles. We have some found in mammals as well, going on and on. So these elements, in contrast to this SHOX gene, these elements are arising at different times on different branches of the tree. So we can basically tabulate this, and that's what we did around the whole genome. We took all 200 of these feather genes and all of the non-coding elements around them, and we basically tallied them up and asked, when did they arise in evolution? So what you can see here is a graph where we have on the x-axis sort of a timeline going from the bony fishes on the left to birds on the right. And on the y-axis, we have a measure of how often these new genetic elements, whether genes or regulatory elements, are arising. Now, we know feathers arose pretty much around 220 million years ago in the most basal dinosaur lineages. And we can, when we look at the actual genes, we find that, in fact, most of the genes required to make a feather were already present even in fishes. We humans have basically the entire complement of genes to make a feather. I sometimes wish if I just closed my eyes, I could just sprout, you know. So genes are quite old, and in fact, you know, many of these genes predate the actual origin of feathers. There is one group of genes, the beta-carotenes, which did proliferate and duplicate in the ancestor of birds. That's what sort of feathers are actually made of. But that's one gene out of 200. Now, when we look at the non-coding elements, we find a really interesting pattern wherein we see one spike not when feathers arose, but actually when tetrapods moved on to land, when amphibians were evolving into the amniotes, as we call them, the mammals, birds, and reptiles. And there was presumably a lot of modifications of the integument, the skin. We also see another uptick precisely where feathers originated. And so these regulatory elements are showing a lot more temporal concordance with the origin of feathers than are the genes underlying feathers. And for this reason, we proposed that, feathers likely had a big contribution of simply changes in gene regulation of genes that have always been there doing other things. And so this is, I think, a hypothesis that you could test with additional molecular tools. Good. Let's take our last example and talk about the loss of flight in birds. And here we're going to focus on a clade that is probably, the least amenable to scientific experimentation. We're always, we folks that work on sort of offbeat organisms, we're always, stressing over the fact that, well, we can't do genetic crosses in our lab, we can't even raise them in captivity, we can't breed them in the lab. And so we wonder, well, how can we effectively study species like this to learn about their genetics? And so we're talking species like the ostrich in Africa, emus and cassowaries from Australia, rheas in South America, kiwis in New Zealand, and then this diminutive lineage called tinamous. Tinamous are a group of about 50 species. They're neotropical birds. They are clearly part of this so-called Paleognathus clade. Paleognathus means ancient jaw. It's basically a particular configuration of the bones on the upper jaw or the palate. Tinamous clearly have this configuration, and yet they can fly. And so they are sort of honorary Paleognaths, but I'll tell you how they play a really important role in our interpretation of the loss of flight in this clade. Now, flightlessness is it's a multi-dimensional phenotype. There are many aspects to a flightless phenotype, one of which is loss of the keel. So this is the sternum. Most birds have a really big outgrowth of the sternum called the keel, which is basically where the flight muscles attach to. The ratites, which are the flightless paleognaths, they've lost this keel, and in fact, that's why they're called ratites, because their sternum looks like a raft. There's also big changes in body size in many of these birds. You know, these are some of the largest species to have ever lived in terms of birds. They also include, for example, the moas of New Zealand, that major group of birds that was driven to extinction by the arrival of humans, and also the elephant bird from Madagascar, which was one of the great, one of the largest birds to have ever walked the earth. Of course, there's also a lot of skeletal changes in the forelimbs and in the hind limbs as well, but particularly the forelimbs, where we see loss of or reduction of the humerus, radius, and ulna, as well as many of the digits. We've all seen that ostriches, for example, can use their wings in display, but of course they can't fly with them, and that's partly because their forelimb elements have been greatly reduced. Moas, it turns out, the extinct clade from New Zealand, have lost their forelimbs entirely. We've never found a forelimb from a moa. And so this is one of the key aspects of flightlessness that we're interested in this project. Now, for many years, it was assumed essentially that these tinamous essentially fell outside the flightless clade, and that would be very convenient because it would basically mean that flight was lost once in the common ancestor to the flightless lineages, tinamous being the only flying lineages. And in fact, for many years, researchers basically relegated the tinamous to a so-called outgroup position. They basically assumed that it's the outgroup. It's going to be that first branching species, resulting in a single loss, scenarios involving a single loss of flight. Now, it was only in about 2008 that researchers basically relaxed this assumption, and they allowed tinamous to fall wherever they might in the phylogenetic tree. And remarkably, this is overturning 150 years of our understanding of these birds. Tinamous fell not at the base, but right in the middle of this flightless radiation. And this changed our view of the loss of flight profoundly in this group. Rather than being lost a single time in the common ancestor, we now suspect that flight was lost as many as 6 different times in this clade, all of which is driven by this key phylogenetic placement of tinamous here. Now you could, I know what you're thinking. You're thinking, well, why not have a loss of flight at the base of the group and a re-evolution of flight in the tinamous? I mean, that's only 2 events, right? As opposed to 6. In phylogenetics, we call that parsimonious, the fewest number of changes that can go into an explanation. And the only thing I would say is that, well, it's a lot easier to lose flight than it is to gain flight. Gaining flight, it's only been done a few times, really, in the history of life. It requires a huge set of changes in anatomy and physiology and neurology as well. And if we make some simple assumptions about how easy it is to lose flight, there actually is a pretty good region of parameter space, the rate at which flight is lost or gained, where we might actually expect multiple losses of flight to be more likely than a single loss and a single gain. So here we're just looking at rates of evolution of flight or loss of flight. And we find that it's a plausible scenario if we assume that flight is lost relatively easily. So, and we're going to exploit this because by appreciating that loss of flight is a convergent trait, that it's evolved multiple times, we can make some very powerful inferences about what parts of the genome might be driving it. So we did a lot of sequencing using now sort of second-generation methods, but we got a lot of nice comparative data. We were even able to, we inherited a genome from this extinct little bush moa that our collaborator, Alan Baker at the Royal Ontario Museum, he had, in a really heroic effort, he had basically put together a pretty good draft genome of this extinct species. These fossils are typically about 3,000 years old, and they're often extremely well preserved. You can actually get pretty good high molecular weight DNA and proteins from these fossils. So this was nice because it allowed us to look at another instance of loss of flight. So, we did a lot of comparative genomics. So this is comparative genomics. We're comparing different genomes. This is basically a visualization of just one section of the genome of all of these species here. And some of the techniques we have now for aligning these genomes and basically asking what parts of each genome is similar or homologous to other parts, they're extremely powerful. We can look at insertions and deletions, such as we see here, all these gaps. And we can look at changes in the sequence as well. So all of these red lines here are single base pair changes comparing different species. We can even infer what the genomes of common ancestors might have looked like. And that's what some of these boxes look like. So here is the common ancestor of the cassowary and emu. Here's the common ancestor of chickens and turkeys. So these sort of phylogenetically based alignment methods are extremely powerful. So we basically marshaled a ton of markers around the genome to test this phylogeny that had been proposed, and we were able to confirm it in all its details. Here are the tinamous, okay, again, nesting within the flightless lineages. The only real uncertainty in this group were the position of the rheas. You see the greater rhea and the lesser rhea, both from South America. We resolved them into these relatively tight clade with these relatively short branches. But despite these branches being relatively short, we've tried to break this tree in many different ways, and it's extremely robust. So we're comfortable with using it as a framework to compare, to look at the evolution of different parts of the genome. So we're going to go back to our non-coding elements. And in this case, we were able to collect over 280,000 of these elements sprinkled around the genome. Okay, these are these conserved regions which are not within a gene or a coding region. And we're going to look at how they're evolving in this flightless clade. And in particular, we're going to look at their ability to, whether or not they're increasing in their evolutionary rate. So remember I told you that conserved regions indicate an important function. Well, when we see a gene or a non-coding element speed up in evolution, experiencing a lot of changes in its sequence, that can mean one of two things. It can mean it's lost its function and it's just sort of drifting, or it could mean that it's maybe achieving a new function. It's going to a different place and serving a different role. And so looking at the rate at which these genetic elements are changing is a key aspect of our toolkit. And the exciting thing is that we can do this on a massive scale, looking at hundreds of thousands of these elements. So here's an example of a non-coding element, which is 250 base pairs, not too long. And you can see that in all of the tinamous and all the other birds that can fly, yeah, we've got a penguin in here, but it flies in a different way, just underwater. You can see how that element is very, very conserved. There are very few of these red tick marks, meaning its sequence is basically unchanged over 100 million years of evolution. However, when we look at some of the flightless lineages, we see a detectable uptick in the number of substitutions, substitution being a change in sequence in these flightless lineages. And so this basically inspired us to develop statistical models to really be able to measure this precisely. And I am not going to walk you through all the crazy math. I want to I do want to show this just to disabuse anyone in the audience who thinks that evolution is a sort of a speculative science. We can marshal all the computational tools and mathematical tools, and it's actually been really exciting to collaborate with statisticians who are thinking about these different models and how we can test them. Bottom line is this is giving us a very sensitive way of looking at rates of evolution, specifically in the context of convergent evolution, these target lineages in red. Previous methods, it's very difficult to distinguish an element that's accelerating on just these target lineages from one which is accelerating across this entire clade. It was very difficult to do that. Our Bayesian methods really allow us to do that with high sensitivity. So we basically scan the genome looking for elements like this, where, again, through 100 million years of bird evolution, these green branches, this element is basically remaining the same, but has accelerated specifically in the two rhea species and in the moa. This tells us that this element might well have something to do with loss of flight. I mean, after all, the only two species in our data set where it's accelerating are these two flightless species. So we can sort of scan the genome and look for signatures like this, and I just love looking at these. You can see all of these elements are accelerating specifically in a subset of these flightless birds, and so these are going to be our sort of elements of interest that could potentially have something to do with loss of flight. Now, what we did next was to ask where in the genome do these so-called ratite accelerated elements reside? What genes are nearby? What genes could these elements be regulating? And what genes whose regulation might have been changed by their acceleration of these non-coding elements? So here's that data. So we don't worry too much about the axes. The bottom line is that all of these genes here in red and pink are genes which have an unusually high number of these ratite accelerated elements in their vicinity, okay? And what's really cool is that we see a lot of genes that we already suspected might have something to do with loss of flight. So for example, TBX5 has been looked at a lot by developmental biologists, both with regard to reduction of the forelimbs, but also loss of the keel on the sternum. So it's very exciting that we're able to pull this out using a completely independent approach. Other genes like DOC1, and we've got some HOX genes in here, all are highly focused on vertebrate development. And when we ask of this gene set here, what are some of the common functions? We get an overwhelming signal for developmental processes, okay? And so this is telling us that this very heterogeneous set of genes may in fact have a common role in terms of development. And I should just mention now that we did exactly the same sort of method looking for these rate accelerations on the genes themselves. So we asked, are the genes themselves accelerating in rate in the flightless lineages? We did find a few, found about 200. Turns out, though, they don't have any real common functions. It's a very heterogeneous set, and we certainly don't get a strong signal for developmental processes. So this is telling us that the regulatory regions perhaps are capturing more fundamentally the common role of these genes rather than the genes themselves. And so one of the funnest parts of this project is that we've had a chance to work with developmental biologists for whom looking at where these genes are expressed and how they regulate morphology are part of their daily bread. And it's very exciting. John Young here, who's a professor at Simmons College in Boston, he was one of our collaborators, and it's been great to catch up with him here at Woods Hole. Here's an example of two genes which experienced acceleration in the, whose regulatory elements experienced acceleration in the flightless lineages. And you can see that they're both highly expressed in the developing limb. And this is exciting because it says that in this case, these particular genes might be involved with limb development. Another way in which we're able to sort of figure out what parts of the developing embryo some of these genes might be relevant in, and some of these regulatory regions, is by looking at what the genome is doing in those as the embryo is developing, and whether or not it's basically accessible to gene expression. And for that, we're using an approach called attack-seq. I won't go into too much detail, except basically what it's telling us is what parts of the genome are tightly wound around the nucleosomes and basically inert in terms of their activity, versus those parts of the genome which are open and available for interaction with proteins to drive gene expression. And the reason why this is important is because, basically, organisms have a single genome, right? But in truth, that genome is different in different parts of the organism as it's developing. And so, for example, we can look at whether we have these so-called open chromatin regions in the developing forelimbs of this chicken. Remember, it's one genome, but it's got a very dynamic interplay of being active or inactive. And so we did that for a bunch of different tissues in developing chickens. And what we found was that, so what this shows is that these non-coding elements that we've discovered are overrepresented in regions of so-called open chromatin in these various developing parts of the embryo. Here we've got a forelimb, hind limb, keel, flight muscles. We're finding that these non-coding elements are often found in these regions of open chromatin. What that means is that they are likely genuine enhancers. They're in regions that are actively transcribing genes. So this was a big discovery for us because it showed us that these markers whose function we didn't really know about, not only now do we know where and when in the embryo they might be active, but we can basically fundamentally establish them as true regulators. It's something we can't really know just by looking at their sequence. And so we basically have been looking around the genome for situations like this, where we have an element here in, say, a chicken, which is clearly an open chromatin. What these little peaks mean that that part of the genome is highly active. So this is sort of an active enhancer. But in the flightless rhea, for example, that region is inactive. So this is showing us how epigenetics is could be changing the deployment of genes, and maybe one of the mechanisms by which loss of flight occurred. And so we basically took several types of evidence to really hone in. I mean, remember, the genome is huge and complex. We got to distill it into a short list, a short candidate list of genes that might be important and regulatory elements. So we basically took our data from rate acceleration studies, from attack-seq, and also from other kinds of epigenetic marks, and came up with a very manageable short list of elements. Now, we're not claiming that these 42 elements would be responsible for loss of flight in these birds, but it takes, we started, remember, with 280,000. So this is really allowing us to take on a very manageable set to look at. And what we can do now, with the help of folks like John, is to ask whether these elements can indeed drive gene expression in the developing limb, because that's going to be a requirement for any role they might play in, for example, forelimb morphology. And what these experiments show here is our interrogation of one element that was accelerated in some of the flightless species, and we're asking whether that element can drive gene expression in developing chick embryos. We can look at the chicken version. So this red dye here indicates that we've successfully injected this construct. I mean, we're basically trying to make a flightless chicken here. We've successfully injected this construct, and this green shows that, yes, it is indeed driving gene expression. Now let's take the tinamou version of that same element. Yes, we've injected it, and yes, it continues to drive gene expression. So this is looking like a flight enhancer, perhaps. When we take the rhea version of that same enhancer, we can inject it clearly, but it doesn't show any evidence of being able to drive gene expression. So this is one piece of evidence suggesting that elements like this, enhancers like this, might have played a role in loss of flight. And it's really experiments like this which are convincing us, like the origin of feathers, that loss of flight is much more a function of gene regulatory change rather than change in the actual sequence of genes. And what's really exciting now is we're working with folks like Emma Farley, professor at UC San Diego, to not just look at a handful of these enhancers, but we're taking several hundred enhancers at the same time. We're able to sort of, in a high throughput manner, assay several hundred of these enhancers and find out which ones, in fact, are driving gene expression in various organs. And so this is something that we're actively working on right now. So hopefully I've convinced you that comparative genomics can be a very powerful tool for looking at links between the genes and the phenotype, even for very untractable clades like flightless birds. And so our hope is that the model we provide with these flightless birds will be an inspiration for other biologists to look at their intractable clades. And so I just want to end with a list of our collaborators and to look forward to your questions. Thank you very much.
All right, let's get it going. Probably not going to have time for it.
Thank you. That was an awesome talk. I'm curious about the interplay between regulatory and gene sequence changes. So is there any sort of temporal component where you're exposed to certain pressure for a certain amount of time? Generally speaking, it'll first be a regulatory change, but it will eventually yield to a sequence change? Or are there different examples where you can predict which one will happen given a certain trait?
That's a really great question. And the bottom line is we're still in a phase where we're picking traits piecemeal and probably looking at a lot of the low-hanging fruit. And so a lot of the first studies to find these links in non-model species, we're looking at things like coat color in mice. Is it melanistic or not? These are protein coding changes, and we can find the gene that's responsible. More complicated examples or continuous traits is something getting bigger or smaller. That's going to probably be many genes, but also a lot of gene regulation. So what we need now is to sort of amass a list and just figure out what's going on. Is it maybe a lot of the continuous traits are regulatory, whereas some of the more discrete traits might be more protein coding chains. But even there's a lot of different things that could go on. And so we're still in that. We're accumulating examples, and so we're not yet in the meta-analysis stage. Yeah, great question.
Thank you. That was a great talk. I enjoyed it very much. I'm taking a similar approach to understanding gene regulation during regeneration. The question I had for you is, for those conserved elements, what's the size range that you're looking at, and what is the limitation of how small you can detect those, the conservation?
Yeah, that's a great question. I mean, we focus just on elements greater than 50 base pairs. That's going to be a minimum for detecting rate changes, for example. I mean, you might have a 10 base pair thing, but rate changes are going to be very difficult to detect there. But some of these elements get up to several thousand base pairs, and we can look at a lot of them encode these motifs essentially that are bound by transcription factors and stuff like that. So I think it's much easier to find a signal of acceleration.
So what's the longer? So the minimum is 50, but what's the maximum of these conserved regions that you find?
I mean, I'm sure there are enhancers out there that are tens of thousands of base pairs. It depends, and of course we're still defining them, and so we have a set that we know kind of acts as enhancers, but there's probably a lot of more complex ones that we still have to characterize and define. So.
And just a follow-up question. do you ever detect, so you're looking for them sort of based on their location in relation to other genes. Do you ever find conservation of these enhancer sequences that may have popped over in different regions that may have gotten misplaced or so? Um,
Mean displaced in terms of,
as there are rearrangements going on
well,
across
yeah,
evolution.
and that's, I think, one of the challenges of this study is that, we can't necessarily assume that the gene next door is going to be the target of regulation. And so I think there's a lot of cases where that will be the case, but as I mentioned, we've got these phenomena of long distance regulation where an enhancer might be controlling a gene, hundreds of thousands of base pairs away. And so now there are approaches, looking at sort of the 3D topology of DNA where we can figure out perhaps some of the genes that enhancers are actually regulating. So yeah, I think that's definitely an important thing for the future.
Hi, you fantastic talk. This was really fun. I grew up here. Okay, so my first question is statistics. Are we talking? I'm an astrophysicist, so are we talking 3 sigma results when we're talking numbers here?
Talking what, sorry?
3 sigma?
3 sigma.
Yeah. So on the student F tests, yeah, no, okay.
You mean standard deviations or what?
Are we like when you the report of the numbers are 99.5 or?
You mean statistical significance?
Yes,
Or-
exactly.
Yeah, we're using an approach called Bayes factors.
Exactly. So you're using Bayesian analysis.
Yeah.
Okay, check. Okay, now I have a fun question. What happened to the dodo bird?
It's a flightless pigeon.
Okay, so where does it live on your ladder?
Well, it's a pigeon, so we didn't look at it
No,
in
we
our
didn't.
analysis. But that's a good point. I mean, I think what we'd like to do is take some of these enhancers and try them out on other flightless lineages.
So
I think that would be really
why
cool.
did it die?
Why not?
Why did it? Didn't it die? I don't know.
I mean, we're on it. We're working on it. Give me a few years. It'll happen.
Fascinating talk. So you discussed a lot evolution of flight and feathers and so forth, and the way you described it was kind of this binarization, like either they're flying or they're not. And I wanted to ask, and you kind of implied a little bit about, I don't know, presenting and so forth, but I guess evolution has to operate continuously, and so how do you think about that for something as complex as flight, which requires considerations of weight and aerodynamics and so forth? I mean, how do you really think about that? I'm not an evolutionary biologist, but the first thing that comes to mind is like a flying squirrel, where you can.
No. Give yourself more credit. This is. I would hope my graduate students would ask questions as good as that. It's a very good question, and yeah, actually, that's the focus of our recent NIH grant is to develop models that will allow us to look at these so-called continuous traits. Because you're absolutely right. I mean, to call a chicken flight capable is giving it way too much credit. Right? If you had a graded system, chickens would probably be pretty near the bottom. As would tinamous. Tinamous, they're one of these burst flyers, just like chicken type birds. They can burst and fly for like 30 or 40 yards, then they fall down and they're done for the day pretty much. So can you call that volant, or is it So I think characterizing phenotypes in this continuous way is going to be really important, and I think. And it could be more challenging as well. And so we're probably going to need hundreds of species in order for this approach to be really effective. Yeah.
Unfortunately, I have time for just one more, but I'll remind you that you can join the speaker at a reception just following over it. So.
Hi, I have a practice of science question. So not really, it's about things that you alluded to in your talk, but not necessarily the research. But biology is a ratchet, and then biological publishing is also a ratchet, where as you have these large hypotheses encompassing a large variety of species, you need more evidence to support it. Compared to the paper that you alluded to in your first slides of your talk on this one piece of data being the 1% difference between chimpanzees and humans, and you're able to have this huge hypothesis about, okay, this is gene regulation. If you wanted to make that claim nowadays, you would have to have like an ATAC-seq and whole like gene regulatory like profile of all these species in order to say that. How do you think this change in biological publishing has changed your approach to science and also the rate at which you propose hypotheses and ideas for your work?
You mean just sort of becoming more comprehensive or involving more tools or?
Yeah, with this phylogenetic approach, you had to take samples from hundreds of species,
Yeah.
and so it's a kind of a different approach than has biology been traditionally practiced.
Yeah.
I'm wondering
well-
how that shapes your collaborations, and also do you think that that has impeded your ability to communicate your science faster, or how do you think it has impacted the way that you do science?
I mean, I think collaborations, they take work, and and the hard part is finding collaborators that you can get along with and that see the world as say, trust me, I've gone down lots of dark alleys in that regard. So I think you mentioned the speed at which you can publish stuff. I mean, one of the things I learned as a graduate student, which I've always taken to heart, was a postdoc at the time said, good science takes time. Now, the challenge, of course, is that there are pressures to get tenure and pressures to, publish the least publishable unit. But honestly, I think a lot of people here appreciate that, exciting science is going to be integrative science where we're bringing in lots of different tools. And so, I would hate to think I think the publishing world does have an undue influence on scientific practice, but, well, when you're at my stage, you don't worry about that stuff. So I think my goal is to try to produce the most integrative work I can, and I'm not as worried about where it might eventually appear. So, yeah.
Well, you can throw that influence around, though. So let's thank Dr. Edwards, and please join him at SWOPE.